Hi, my name is Bezagi Tacho, and I'm an assistant professor of civil and environmental engineering at Rice University. Today, I'll be sharing the work we've been doing in my lab on active fouling removal from electrically conductive membranes with an interdigitated pattern. The work I'll be sharing with you today was primarily done by Subin Cho, who is a fourth-year PhD student in my group, with support from other lab members. So membrane-based processes are an important and growing part of the water treatment industry. One of the persistent challenges in membranes is fouling. So the deposition of organic material and bacteria, the growth of bacteria and biofilm on the surface, and the formation of inorganic scale reduces water flux through membranes. To combat this, backwashing is done in low pressure membranes, and there's also chemical cleaning that takes place to more thoroughly clean membranes. A recent study reported that fouling accounts for about 10 to 24% of the operational expenses in membrane plants with relatively clean sources of water. Now, this is likely to be higher with more complex feed water. In a lot of cases, membranes are not considered a viable option if the source water has very high fouling tendency. As an alternative or a supplement to current cleaning strategies, researchers have been exploring electrically conductive membranes. So with a conductive membrane, the surface becomes a dynamic material that its charge can be easily changed, and it can also run electrochemical reactions that are effective for not only reducing fouling, but actively removing it once it forms. So for example, when negative potentials are applied, the membrane surface can repel negatively charged foulants, or water splitting reactions that you can see in the bottom left of this graph here can generate hydrogen bubbles that actively scour or remove things that have adhered to the surface. A change in the pH at the surface that accompanies the water splitting reaction can also help dissolve some types of inorganic scale. On the anodic side here, there can be direct or indirect oxidation of organic foulants in bacteria that reduces their attachment and can actively remove them. Oxygen evolution reaction can also scour the membrane surface, or it can dissolve scale that is more soluble at low pH values here. And overall, electrochemical fouling control using a combination of these mechanisms has been reported to be much more energy efficient than cleaning biochemical additions or the other current strategies that are used. So one of the challenges in implementing this idea is how to practically integrate two electrodes in a membrane module. If the membrane surface is being used as the working electrode, there needs to be a counter electrode, which can be a separate rod or a sheet or a permeate spacer. In some studies, there's another electrode on the permeate side that serves as a counter electrode. Additional challenges or questions that are present are the stability of electrodes over long-term operation, damage to polymeric membrane active layer from electrochemical reactions, and lower membrane flux due to the added conductive layer on the membrane. So in my lab, we've been exploring the idea of co-locating both the working and counter electrode on the membrane surface as a way to overcome the limitations I just mentioned. Particularly for spiral wound modules, this would make it easier to integrate into existing modules. In addition, if sufficient fouling removal can be achieved with only partial coverage of the membrane surface with an electrode, kind of like how defroster grids on the back windows of cars work, this can help with the other challenges I mentioned as well. When we did the literature search on this idea, we found one paper that reported the performance of microfiltration membranes with interlaced or interdigitated carbon nanotube electrodes. They looked at the difference in bacterial fouling and backwashing efficacy with these membranes, and they found lower bacterial attachment on the membranes with mixed results for backwashing efficacy. The mixed results for backwashing was mainly because the conductive membrane had lower fouling to begin with. We thought this study was promising, but the surface coverage was fixed at 80%, and we were interested in providing a more systematic understanding of how electrochemical fouling removal works as a function of surface coverage. So to provide this more systematic understanding, I will share with you the results from in the next couple of slides from a study we published in the SNT Engineering in 2023. So in this paper, we wanted to test how changing the level of coverage with an electrode alters the amount of biofilm that's removed from the surface electrochemically. Is it going to be a linear relationship or a nonlinear one that plateaus at a certain point, or some other unexpected relationship? And so that's what the heart of this paper was about. So this was the general outline of the experiments that Subin did. She made a series of patterned electrodes using laser scribing on a polyimide support. She grew biofilm on these electrodes, and she used fluorescence to quantify the amount she'd grown. Looking at the fluorescence on the different electrodes let us know that there was no systematic increase or decrease in the amount of biofilm grown as a function of electrode coverage. Finally, she applied a range of potentials to electrochemically remove the biofilm and compared those values. Interestingly, what we found was that fouling removal, as measured by a decrease in fluorescence intensity, as well as staining by crystal violet assay, although that data is not shown here, it increased at first and then plateaued at around 53% coverage. This trend was the clearest for the highest potential we tested, which was 2.5 volts. These results are for the cathodic condition, which was generally more effective than the anodic condition. So this result overall shows that sufficient fouling removal can indeed be achieved without full coverage of a surface with an electrode. One of our hypotheses about the mechanism behind this trend was that there's a minimum amount of bubble generation that is needed to scour the biofilm fully. And once that is met, any extra current that we supply is not really needed. To see if this was the case, we looked at the current that was generated on the coupons, and what we were expecting was for this number to become more negative as the electrode coverage increased, which, as you can see, it does for the surfaces without any biofilm in them, the whites or the white circles here. But surprisingly, for the biofilm covered electrodes, which is the red data points here, the current was actually pretty steady as a function of electrode coverage. So this suggests to us that the shape of the electrode may play an important role in a removal, since 53% is when we start to have these electrode arms coming out in the pattern. And that's something that we plan to look into in the future. The resistance and electrochemical activity of the pattern depends on the amount of carbon nanotubes that is deposited. We found that a carbon nanotube loading of around 1 milligrams per centimeter squared was needed for sufficient current density, as you can see on this graph here. And this was also the loading where the resistance did not change much as a function of the width of the conductive lines. Using SEM images, we can see that at this carbon nanotube loading, the thickness of the layer was around 18 micrometers. So after making membranes with sufficient conductivity, Subin measured the membrane-specific flux in a cross-flow filtration system, as you can see in the schematic here and the images here. She then fouled the membranes with a sodium alginate solution until the initial flux decreased by about 80%. After fouling, a potential of 4 volts was applied, alternating the polarity every 30 seconds for a total of 3 minutes. The flux at the end of 3 minutes was recorded, and we calculated the self-cleaning efficiency using the equation at the bottom right here. Interestingly, the level of self-cleaning with the patterned membranes was very different from what we saw with the patterned surfaces, as you can see in the results here. For the range of the conditions tested, self-cleaning was between 20 and 60%, with only a few of the pairwise comparisons having statistically significant difference. There are several major differences between the membranes and the surfaces. First, self-cleaning is measured differently. Here we're looking at flux recovery versus using fluorescence to see the amount of biofilm. We're also dealing with a different type of fouling with the membranes versus the surfaces. In this case, both in the membranes, both anodic and cathodic reactions were taking place. And finally, the amount of fouling that's deposited is likely very different between the two studies. And particularly with the membranes, it might not be homogeneous across the membrane, and we might have preferential fouling on the non-conductive areas where cleaning is limited, whereas cleaning is limited to the conductive areas. So some of the complicating factors for understanding the results with the membranes are that, one, there's a large difference in permeability of the base polyether sulfone membrane and the carbon nanotube layer. The polyether sulfone has a very open and porous structure, as you can see in the SEM image here, and the CNT layer has much smaller pores. And you can see this difference in the water flux through the polyether sulfone membrane, pure water flux, and a membrane that's fully covered with the carbon nanotubes. So this might introduce some differences in the amount of fouling that occurs in these two regions. SEM images of the polyether sulfone regions that are shown here show that there's clear fouling after the sodium alginate solution is filtered through the membrane. And there's also some fouling remaining on the surface there after cleaning. It's more difficult to differentiate or clearly see the level of fouling on the carbon nanotube regions, which are the images shown here. But similar to the polyether sulfone, we see what are likely sodium alginate particles on both the fouled and the cleaned membranes. So these SEM images that I showed in the last two slides suggest a few things about what could be behind the limited self-cleaning. First, the alginate fouling is not a continuous film, but it's an isolated kind of clusters, which is different from the fouling we had imagined would be taking place. Secondly, the relatively thick CNT layer here means that bubbles that are generated at the CNT surface are unlikely to reach the polyether sulfone region and scour that area. There was work in the literature that reported near 100% flux recovery from scouring from conductive feed spacers, which had led us to believe that the bubbles from the conductive regions could effectively clean the non-conductive area. However, after our results, we went back to that paper and noticed that they used sodium chloride as the electrolyte, which means that there might have been a significant level of indirect oxidation that contributed to that level of self-cleaning. And in fact, we saw that when we changed the electrolyte to sodium chloride from sodium sulfate, we did see a significantly higher levels of cleaning, although we don't yet have the data on whether this is from enhanced cleaning of the CNT regions only or both the CNT and the polyether sulfone regions. A couple of other findings from this paper are that the pattern parameters, such as the electrode width here and the spacing, do impact current density. So here current density is the measured current divided by just the electrode area and not the total membrane area. We found that at constant electrode width, so the top two graphs here, smaller spacing increases the current density at constant spacing. Sorry, at constant electrode width, smaller spacing increases the current density. And the bottom two graphs here, at constant spacing, smaller arm widths result in more current. So this could be used in the future to improve the energy efficiency of electrochemical fouling removal. Another interesting finding from our results is that where the fouling takes place, meaning whether the fouling is taking place mainly on the surface or inside the pores, has a big impact on the level of cleaning that's observed. So when we use a lower concentration of sodium alginate here, or the blue line here in the feed solution, we can see from TOF-SIMS mapping that more fouling takes place inside the pores, and fouling also takes longer, as quantified by the membrane fouling index. In these cases, the self-cleaning efficiency was actually negative, meaning that the flux continued to decline after applying current. So this is an important finding that we would also like to highlight, and has implications for electrochemical fouling removal. Okay, so to summarize and conclude, we measured biofilm removal from interdigitated electrodes and organic fouling removal from membranes with interdigitated electrodes. Our results from pattern surfaces indicate the potential for effective biofilm cleaning with 50% surface coverage, but results from the pattern membranes point to the need to reduce the electrode thickness, match electrode permeability with base membrane permeability, and make use of a range of electrochemical reactions to get higher self-cleaning efficiency. With that, I'd like to thank my group for doing this work and just being a wonderful group of people. And I'd also like to acknowledge our funding sources for the financial support to pursue this research.